All right. I think we're good to go. Isabel, off to you.
Thank you. Welcome, everyone. It is my pleasure to welcome to this last seminar series for the fifth consortia. So looking forward to hear from the Atlantic Cancer Consortium. We are accompanied by members of the Network's Patient Working Group, as well as researchers, clinicians, trainees, and leadership from all the different consortia. We are also joined by some of our funding partners, including the Terry Fox Foundation. I want to congratulate everyone on their tremendous work and effort to successfully deliver on phase 1. With only 4 more months to go, we are doing an amazing job on building the gold cohort with almost 13,000 out of the 15,000 cases already sequenced. And I also want to share and acknowledge that the great news that the funding for phase 2 has been approved and is in the general in the budget from government. So really great news, really exciting times that we are heading towards our phase 2. And I will leave it now to Veronique to present this seminar that will be hosted by the Atlantic Cancer Consortium, as I said, and I know that we have a packed agenda. Thank you, Veronique, for your tremendous work with this seminar series.
Thank you very much, Isabel. Welcome, everyone. I'll start off with just a little bit of housekeeping. We will be keeping questions until the end of the seminar. So please either type your questions in the Q&A box that should be at the top of your screen. It's 2 little speech bubbles with a question mark in it. Or you can wait until the question period and raise your hand. You can also do that with the little hand icon at the top of your screen. And then once we get there, we'll allow you to unmute yourself and you can ask your question live. Before we get into it, I'd also like to take a moment to acknowledge that I personally, as well as a few of our speakers today, are calling in from Mi'kma'ki, the traditional and unceded territory of the Mi'kmaq people. Our network members and some of our speakers and our attendees today are located all across the country, as you know, on lands that are home to diverse First Nations, Métis, and Inuit populations. So I invite everyone attending today to recognize and to think about that and to honor those peoples and reflect on the importance of those lands. I would also like to introduce my co-chair, Natalie. Natalie is an inaugural member of the network's patient working group, and she's also a member of Network Council. On top of all the work that she's doing and that the whole patient working group is doing, she generously agreed to co-chair her second seminar with us, actually. So Natalie, thank you so much for joining us, and I'll pass it to you if you'd like to start us off with a little bit about yourself.
Awesome. Thank you. Sorry, my dog was barking. Hopefully he stops. My name is Natalie Quadrans. I now call myself a Calgarian, although I was born and raised in Montreal. Did my Bachelor of Science at Concordia University in the mid-1990s. Now that makes me old. I moved out west to compete with Team Canada's snowboard racing team between 1995 and 1998. Then fast forward a couple of decades, I accumulated a few degrees in between. I got my MBA in 2000, my CMA CPA designation in 2011, and I was halfway through a Master's of Science with HEC when I was diagnosed in 2019 with my de novo stage 4 triple positive breast cancer, because that's a long diagnosis. My kids were only 2 and 5 at the time. My cancer has certainly come with its set of challenges, as most people's do. Our goal for me was to basically remain on my first line of treatment for as long as possible. We played a lot of whack-a-mole, radiating my spine in 2020, my hip in 2021, and when my cancer returned for a second time in my hip, radiation wasn't an option, so I was lucky enough to be able to join a study that allowed me to get a full hip replacement, and that gave me 2 full years of progression-free cancer. Last May, it came back again, forcing me to move to my dreaded second line of treatment on an aromatase inhibitor. Now, last month, I found out that it has come back again, though this time it's in unusual spots for breast cancer. So I have a really urgent PET scan scheduled, which doesn't come till December 12th. As a patient, that's a long wait. And I'm at a junction in my cancer, I hate the word journey, where guidelines now will no longer allow me to look at hormone therapies as a next option. So unless we can resect or radiate, I will be forced to move on to an antibody drug conjugates for the rest of my life. And the thought of losing that quality of life when I'll no longer be able to snowboard with my kids, it terrifies me. Because I still snowboard very slowly. And that brings me to why we are here today. And it's to talk about the research to accelerate precision oncology for patients such as myself. So just a little heads up, all those scientists here joining us, no pressure. But before kicking it off, I did want to introduce Dr. Robin Urquhart, who will be talking to us about where the consortium has been doing. Dr. Urquhart's an associate professor at Dalhousie University. She is also the Canadian Cancer Society Nova Scotia Division endowed chair, there we go, in population cancer. She's a senior scientist as well with the Beatrice Hunter Research Institute and an affiliate scientist with the Nova Scotia Health Authority. So if that wasn't already enough, Dr. Urquhart co-leads the MOHCCN's Atlantic Cancer Consortium, which is what we are here, and she'll be giving us an overview of what activities have been taking place. So with that, Dr. Urquhart, I'll let you take it from here. Thank you.
Thank you, Natalie. Thank you for sharing your story. I'm really, I'm sorry to hear about everything that's happened in the last month or so since we saw each other last. Okay, I'm going to share my slides. This will be very quick because we want to get into think about the science and talk about the science and hear about the science. So I'm going to speak just for a few minutes about the Atlantic Cancer Consortium here. Next slide. I did want to, let's see. I'm just figuring this out, folks. Here we go. I did want to just acknowledge, again, where I'm seated today and where I live and work and play, which is on the ancestral, unceded, and unsurrendered territory of the Mi'kmaq Nation. So I wanted to acknowledge that, and I certainly have the privilege and the pleasure of being in this part of the world. That's a picture of Kejimkujik, which is in interior Nova Scotia, and if you are ever in Nova Scotia, it is simply a lovely place to visit. I also want to recognize the histories, contributions, and legacies of the African Nova Scotian people who have lived here in Mi'kmaq for more than 400 years. Okay, so I'm going to talk very briefly about the Atlantic Cancer Consortium, give just sort of a broad perspective for everyone before we get into a couple of scientific presentations. As you would know, we're comprised of 4 Atlantic provinces here on the east coast of Canada. And I have a picture there of Harry Fox when he started his Marathon of Hope. I think everyone here will know he started it in Newfoundland and Labrador in 1980. And so I'm just going to give some context about this area of the country. This is, I'll tell you what this is. This was just released this week. It is the latest map of Canada around age standardized incidence rates from the 2025 Canadian Cancer Statistics. So for many of us, at least for me as an epidemiologist, this is sort of a very important report. And so you can see here, it doesn't look any different than we've seen for years. There's a bit of a, west to east gradient in terms of incidence rates for cancer in our country. And you can see here that the Atlantic provinces as well as Quebec really are projected to have the highest incidence rates this year with all cancers combined. This is from the same report, again, just released on Monday. And you can also see here that in terms of mortality rates, the eastern provinces or the Atlantic provinces, along with Nunavut, have the highest age standardized mortality rates. So we in Atlantic Canada are sort of find ourselves in this situation often where we have, for most cancers, very high incidence rates, age adjusted, age standardized, and very high mortality rates. Nonetheless, it's a beautiful part of the country to live in. But there are a few other things that I wanted to point out in the context of where we live and work here and the work that we're doing in the Atlantic Cancer Consortium. Not a lot of people live here. These are the latest estimates from each individual province from 2025. So you can see we're a pretty big region with not a lot of people. Interestingly, a lot of the people who do live here live in rural areas. So depending upon the province, we're anywhere from 41% rural population to 53%. And of course, that's more than 2 times the national average. And so that has a lot of implications when you think about accessing care, right? From the health system's perspective for delivering care, and then from like patients and families' perspective to actually access care. We have an older population, broadly speaking, than the rest of the country, and we all know what that means when it comes to cancer. The latest Statistics Canada data that has come out around poverty in Canada shows that both Nova Scotia and Saskatchewan are amongst the provinces with the highest rates of poverty. With some more recent data from 2025, not from Statistics Canada, from another source, does show us that about half of adults in Atlantic Canada are less than $200 away from insolvency. And I bring that up because I think that's really important in the context of getting a cancer diagnosis. If you are less than $200 away from being able to pay your debt and to be able to pay your bills, that's a really important thing. And again, the latest data show us that in this part of the country, along with Quebec, about 30% do not have a primary care provider. Of course, lots of implications there as well around accessing cancer care and early diagnosis and so on. This is a quote from a patient partner who started with us from the beginning in the Atlantic Cancer Consortium. And this came from one of my first conversations with her. And she said to me, as we 6 or 7 years ago, my biggest fear when I was diagnosed was that I would not have access to the same treatments because I live in rural New Brunswick and not downtown Toronto. So this is a real fear for patients in this region. And I would say this is one of the reasons why I'm so passionate about the work that we do. And I don't have a lot of time to talk about that, but I really think making sure that what we do is accessible to all Canadians, regardless of where they live, regardless of their circumstances, is the most critical thing that we can do here. Okay, so from the beginning, I'm not going to get into this, but just tell you a few highlights. We started in our sort of early phases and sort of pilot phases in 2020. That took a couple of years. It was extended because of the pandemic and all kinds of things. But essentially, a couple of the highlights was we really had to build our biobanking efforts here in Atlantic Canada. And so we were able to we have built 2 biobanks from scratch just in the last 5 years and have expanded 2 other biobanks. So I think that's an incredible testament to the work that has been done here. And we ended up spending a lot of time early on in terms of training and capacity building. So we supported 22 trainees, postdoctoral fellows, grad students, medical residents in precision oncology. And that's really important to us here in Atlantic Canada from the perspective of building capacity. And we had a number of sort of projects that we were working on together to, work together, demonstrate we can work together across provinces and so on. Right now, so we became sort of this full-fledged member in 2023 in January. Here are some of our core themes or core goals, I guess. One is certainly contributing to the gold cohort and contributing about 1,300, just over, cases to the gold cohort. And then we have these other themes here. We're doing a lot of work around liquid biopsy, genetics, and environmental interactions, which is a big area of focus and area of science in this part of Canada. Genetic drivers of cancer, tumor microenvironment, metabolism, and a focus on access and equity. So today, this is, I think, my last slide. Today, here in Atlantic Canada, we have 10 member institutions, And the latest that I have anyway, and I think this is pretty much up to date, is that we have 114 individual members who are contributing to this. And I think that is amazing for Atlantic Canada. Like, I really think this, as I say, warms my heart that we have so many people invested in this initiative here in this area of, again, Canada. To date, we have fully sequenced 1,000 cases, which is fantastic. Yay to all of us here. And we have just under 400 of those cases have their full clinical data contributed. And we are on track to meet these by the end of the year. I did want to point out something, and I'm just about done. It's very interesting here in Atlantic Canada to think about how all of this happens. This is one example. So, and I say all of this in terms of the sequencing and generating that gold cohort and then doing research. So if you're in Atlantic Canada, let's say we take a case from the QE2 Biobank, which is in Halifax at the hospital here. We first send that to another institution in Halifax, which is Dalhousie University, to the genomics core to do library prep. That's then sent to the Atlantic Cancer Research Institute in Moncton for sequencing. Data are then sent to CARES, a high computing facility in St. John's at Memorial University in Newfoundland for data storage. And then for the research to access those, the data are essentially then sent back to that researcher and back to Dal. So we have a lot of crossing of borders of material and data. And I want to thank everybody that's involved in our initiative and our consortium. For sticking with us to figure all of that out, because it is a lot of work, as you can imagine, to figure out all of those transfers and to do it all in a legal, ethical, safe way. And right now we're going to talk about some of the science. We have 18 active scientific projects ongoing that we're sort of leading, and of course we're collaborating with many others. And I'm going to stop there because we're going to hear about 2 of those projects right now. Thank you.
Thank you so much, Robin. That was a fantastic overview of the Atlantic Cancer Consortium. In terms, like Robin said, we're now going to jump into some of the research. So it's my pleasure to introduce Dr. Anthony Reiman. Dr. Reiman is a medical oncologist at the St. John Regional Hospital, a professor in the School of Integrated Health at the University of New Brunswick, a professor in the Department of Medicine at Dalhousie University, and a senior scientist with the Beatrice Hunter Cancer Research Institute. As you can see, today, Dr. Reiman will be telling us about myeloma research in the Atlantic Cancer Consortium. So Dr. Reiman, over to you.
Thanks. Can you hear me?
Yes.
Okay, great. Well, thank you. It's a pleasure to get together with this group. It's nice to see some friends from across the country have joined to hear about what's going on in Atlantic Canada, and to see so many of our Atlantic team turn out to support one another. Before I start, I'll just say that in addition to the other land acknowledgements that you've heard, in St. John, New Brunswick, here where I live, we're on the St. John River, which is known to Indigenous people as the Willistook River, which means the beautiful river. And this is the traditional and unceded territory of the Wolastoqiyik and Wolastoqey people. So I'm going to highlight research that's happening as part of the Marathon of Hope Cancer Centers Network in Atlantic Canada, focusing on multiple myeloma, which is a bone marrow cancer. And we're going to highlight some research being done that looks at what causes myeloma, how can we better predict the time course of the disease. People are often wondering how long they're going to live with this incurable bone marrow cancer. How can we better understand the biology that drives myeloma? And then how can we apply that understanding to develop better tools for monitoring the disease, to individualize treatment options based on reading the myeloma genomes of each patient, and to see if we can develop new treatments based on our knowledge of this biology. So when it comes to trying to understand why myeloma develops, one of the leaders in our region is Amy Trottier. Amy's a hematologist in Halifax, and I just want to acknowledge all the hematologists who have been contributing by enrolling patients in this program and obtaining bone marrow and blood samples from myeloma patients for our Atlantic Cancer Consortium and for other projects in the region. So Amy is particularly interested in inherited cancer susceptibility, and so she's got a collection of families that have been referred to her sort of familial hematological malignancy clinic. And so she'll have family trees like this, where there's lots of cancer, as there is in every family. But this family history is striking because there are many cases of myeloma and related disorders, and some other cancers in this family. And the question is, why is there a cluster of such cases in such a family? Is this shared environmental exposures? Is this shared genetic predisposition? Some combination of those things. And so Amy's trying to get at this. And so she's got now more probably than 129 patients enrolled in her cohort, but she's obtained germline tissues as well as tumor tissues from patients with malignancy and banked those. These are being subjected to sequencing, many of which are being sequenced through the Marathon of Hope effort. And together with that, Amy and her colleagues are looking at environmental factors that might lead to predisposition to malignancy, including myeloma, banking toenail clippings and looking for heavy metal exposures, looking for radon exposure, and then trying to put together various, genetic loci of susceptibility with environmental exposures to try and explain, what's happening to families like the one in the family tree that you see here, or the two families here that you see. So Amy is also going to lead our effort nationally to contribute to the discussions in the next phase of the consortium on the heels of the great announcement from the federal budget that we're going to have another cycle, and she's going to be our point person on figuring out how to deal with the detection of inherited genetic susceptibility to cancer, and how to work with our patients who have those findings. So when it comes to patients who actually have been diagnosed with myeloma, so this is Manal Alnai and medical student Sam Cutler, who she's been working with in Halifax. Manal runs some of the clinical lab programs in Halifax for patients in the cancer system. But she's got a particular interest in multiple myeloma. And so first project that these two folks have done together, which is focused on understanding myeloma genomics, uses next generation sequencing based technology to analyze the genomes of myeloma patients. They've developed a myeloma specific gene panel. Others have done this, but this is the panel that they've come up with. And what's interesting about this particular panel is it looks at a certain set of genes that are felt to be important in the pathogenesis of myeloma. And if you look at patients who have high-risk genetic markers identified by other more traditionally used laboratory techniques like fluorescence in situ hybridization or FISH, that you can identify high-risk patients with this gene panel that are largely non-overlapping with those that are identified by FISH. And if you take patients who don't have any high-risk FISH markers, you can risk stratify them on the basis of this gene panel into high and low risk groups that have different progression-free and overall survival distributions. So these folks have done some work to help us better understand how to use an understanding of myeloma biology to predict patient outcomes. Sam and Manal have also worked together on some bone marrow plasma proteomics studies. Sam used to work for SEER, which some of you may be familiar with, a company that has expertise in serum proteomics. And so they've studied the extracellular fluid, the plasma from bone marrow samples from myeloma patients in Halifax, and have looked at the highly expressed proteins and signaling pathways. This technology has the ability to reveal, tens and even hundreds of thousands of proteins in a human biological specimen. And these bone marrow plasma specimens are particularly rich with various proteins. And they've been able to do some pathway analysis on these and identify that there's a lot of differential expression of coagulation-related proteins and signaling cascades, and that patients who have the lowest levels of coagulation protein expression appear to have the most inferior prognosis. So this coagulation group 1 in green on the survival curves have lower levels of expression. Manal can correct me if I'm wrong at some point. But so this is really exciting and interesting work that uses a new technology and capitalizes on some local expertise. And so we're working with Manal and Sam now to try and cross-validate some of their work on bone marrow plasma samples and blood samples from our cohort of myeloma patients in St. John. And so we'll be telling you more about that in the future. Here in St. John, Thomas Polno-Canil is a professor in the Department of Biochemistry and Molecular Biology at Dalhousie University. We have a campus of the Faculty of Medicine in St. John, New Brunswick. Main campus, as most of you know, being in Halifax, and Thomas is here. Sawmill is a biobank coordinator here and member of the team. And these folks have been profiling lipids using lipidomics approaches in the serum or plasma of our myeloma patients. And they found in particular that acylcarnitines appear to be differentially expressed in patients who are treated with more intensive therapy. And so our fitter patients appear to have a different lipidomic profile than maybe our older, more frail or less fit patients. And so that's interesting. And they're just starting to delve into what we can learn from studying myeloma lipidomics, which is sort of a new frontier. So it's great to have that collaboration. In my own lab, we're also using blood and bone marrow specimens obtained from patients and in our biobank to profile the cytokine patterns in blood and bone marrow extracellular fluid or plasma of myeloma patients. And this is led by Helmi Alfara, who some of you will know. Helmi presented at the first of these national webinars a few years back. He's developed an enduring interest in NK cell biology and NK cells as they relate to cancer from Jeanette Boudreau, who is a leader in this area at Dalhousie University. And so Helmi moved to our lab from Jeanette's a few years back. So he's interested in this because he thinks that understanding the cytokine milieu in myeloma will inform development of immunotherapeutic strategies. And so he's been looking, first of all, to see if you can get a read on what is the cytokine environment in the bone marrow based on looking at blood samples, which are obviously much easier to obtain and to assess over time. And he's been able to see that these are generally fairly well correlated. And that if you look at individual patients, that for most cytokines of interest, the levels are substantially higher in the bone marrow than in the blood, but the blood can serve sort of as a surrogate for that. And he's also been able to compare cytokine levels in the blood with blood samples from people that don't have myeloma, for whom we aren't routinely obtaining bone marrow biopsies. So that's where having this correlation between blood and marrow is helpful, is we can also compare people without myeloma to those with myeloma and look at the cytokine profiles to get some clues as to what the environment is. And also, using sort of a multiplex approach to profiling cytokines, Helmi's got arguably the largest sort of comprehensive look at cytokines in myeloma patients and how these cytokines sort of interact with one another and how the levels correlate with one another. And so he'll be using this information as he designs novel immunotherapeutic strategies for myeloma. And Helmi presented this at the International Myeloma Workshop this year. I'm not going too slow here, but Amy Wong, also in my lab, presented some work at the same International Myeloma meeting about minimal residual disease assessment in myeloma. And so this is carrying forward on a TFRI-funded national myeloma cohort study that we led, called the M4 study, and which helped form the basis of our involvement in the Marathon Hope Cancer Centers network. And so we've now got longer-term follow-up on these patients. It's still relatively short in the scheme of things, but some of the patients in this cohort have now been with us for over 5 years. And so we're looking at how to integrate assays of assessment of tiny amounts of myeloma that might persist despite highly effective therapy, and to get an understanding of that. So there are various techniques for measuring small amounts of residual myeloma, minimal residual disease in the clinical setting that are being developed. We've been partnering with Rapid Novor in Ontario, who has a mass spectrometry-based approach to detecting small amounts of residual myeloma. And we've been also partnering with labs that have clinical grade flow cytometry-based or sequencing-based MRD, minimal residual disease assays. And in this cohort of patients from across Canada, we've also got selected patients who've had imaging-based MRD detection with PET scans. And so we have a relatively unique cohort that has all of these techniques being applied and integrated, and it gives us the opportunity to see how these things integrate with one another. And here's an example of patients, relatively few patients achieve a negative minimal residual disease status when assessed using this mass spectrometry-based approach to myeloma protein detection in the plasma. But those who do achieve negativity, none of them have experienced an early relapse in the first few years of follow-up on this study, whereas patients who are MRD positive are relapsing at a predictable and steady rate. And you can detect relapse with this type of technology much sooner than you can with conventional methods of monitoring the disease in a clinical setting, often several months prior to clinical relapse being determined. And if you combine some of these methods of minimal residual disease detection in the clinical setting, you can get better prediction of outcomes. We've modeled the prediction of early relapse as a function of the results of these different assays. And we're finding that if we combine the most sensitive bone marrow assay, which is sequencing based, with the most sensitive blood-based assay that we've used in this study, which is mass spectrometry based, that you get the best ability to predict early relapse. And so we think that there's a rational way to combine these approaches, especially in a publicly funded, healthcare system where you might not be able to apply all of these techniques all the time. There's a selective way that you could do this. So this was presented also at the workshop, and we're just getting ready to submit this for publication. We have colleagues who are developing novel methods of blood-based disease monitoring. Locally, we have Keith Brunt from Dalhousie University, who's using a nanoparticle approach to enrichment of metabolites in the blood to detect and describe those. And he's been able to find that you can distinguish myeloma patients from people with other health conditions using this technology. And so we're expanding that work. And colleagues in Toronto, Trevor Pugh, Suzanne Trudell, have presented some work on circulating tumor DNA analysis in myeloma patients, which is quite interesting. And so they published some work online already in the sort of non-peer-reviewed setting, and have presented this at some of the big meetings and have submitted it for publication. So lots going on in disease monitoring. As many of you know, the focus of the next phase of the Marathon of Hope is going to be around returning the results of whole genome and transcriptome sequencing of cancer cases to clinicians. And so in our lab, we've got Luke Weinstein and Selena Oakes working on this, and we're partnering with the BC Personalized Oncogenomics Program to bring their approach to reporting genomic findings of potential clinical significance to patients and clinicians. And so we're going to pilot this in Atlantic Canada. Here's an example of a report for one of my patients with several genomic variants in the myeloma cells of potential clinical significance and with potential therapeutic implications, so that maybe in the future, if these patients start to run out of options, we may be able to think of some new treatment options to consider for them. And this is part of our approach to piloting the return of results to clinicians more broadly in Atlantic Canada. I'm going over time, so I'm going to speed through this, but we're also using information about myeloma genomics to inform new strategies for therapy. This is work initially pioneered by Stacey Grieve in my lab, carried on by Samuel Abagunde and now Will Roderick, basically showing that the Hippo pathway in myeloma is active, which means that the transcription factors YAP and TAZ are inactive in myeloma. And Stacey has discovered that this is principally through methylation-mediated silencing of the YAP and TAZ genes. And Samuel has shown that this is also in part due to upregulation of the upstream elements of the Hippo pathway, including MST1, LATS1 and 2. And so Will is working on studying some novel MST1 and 2 inhibitors as potential treatments for myeloma to try to, switch the Hippo pathway off and allow YAP and TAZ upregulation, which functions as a tumor suppressor and downregulates the MYC oncogene and its targets in these cells. So we're developing some therapeutic strategies like this based on myeloma genomics. And Jackson Weir, who's now a PhD student at Harvard, spent some time in our lab trying to identify new targets for immunotherapy using myeloma genomic data and looking for proteins that are upregulated on the surface of myeloma cells compared to other tissues, and identifying potential epitopes that you could target for immunotherapy approaches using these data. And so we've identified some lead targets, and so Amy and Luke and Helmy are all working together to validate this in preclinical models that we can use to test immunotherapy approaches to targeting these antigens, myeloma selective antigens, so that those patients who maybe become refractory to immunotherapy targeting existing antigens like BCMA or others might have another immunotherapy option. And we're partnering with the NRC labs in Ottawa to generate binders to these lead candidate immunotherapy targets, and hopefully to develop various immunotherapy approaches to these, including, given our interest, NK cell-based therapies, which might be particularly useful in people who have already got exhausted T cells from receiving T cell-based therapies. So thank you for allowing me to go a few minutes over time. I guess you didn't really allow me, I just kind of did, but this is just some of the team here in St. John, some of our funders, some of our participating institutions. And none of this is possible without the Terry Fox Foundation, the Terry Fox Research Institute, and the Marathon Hope Cancer Centers Network. And hopefully we can make some worthwhile contributions. Thank you.
Wow, thank you, Dr. Reiman, for sharing your team's research. It's really fascinating. I now have the privilege to introduce Dr. Hayley Alloway. She's a postdoctoral research fellow in the lab of, and I hope I pronounced this right, Tutati Benergraf in the Division of Biomedical Sciences at Memorial University in Newfoundland. Dr. Alloway will talk to us about the research that she is doing about chromatin profiling and solid tumors. So Dr. Alloway, the mic is all yours.
Thank you very much for the introduction. It's Tutati Benergraf. And if you
sorry about that.
No. Honestly, I correct people with this pronunciation regularly, and I'm not even saying
I pronounced it 40 times last night, and it still didn't come out.
I don't have a French accent, so it doesn't come out right on me at all. If you could just go full screen on the slide. And today, going from the previous conversations, which have been fascinating and very broad, I'll be taking us to a much more narrow perspective and speaking in some detail about my research that I'm conducting with Tutati. I don't have control here. Can someone go full screen with the slides, please? Again, my name is Hayley Alloway. I'm a postdoc. I've been with Tutati's lab for 2 years now. I'm at the Memorial University of Newfoundland. And as with the talks before, I'd just like to acknowledge that Memorial University's campuses are situated on the traditional territories of diverse Indigenous groups. We'd like to acknowledge, with respect, the diverse histories and cultures of the Beothuk, the Mi'kmaq, the Innu, and the Inuit. And so with that, going on to my next slide, if we could forward to the overview. I'm working in colorectal cancer.
Okay.
And I'll see, okay? If you can go back to title slide real quickly. Just got a little tripped up there. So my project is specifically refined ChIP-seq protocol for high quality chromatin profiling in solid tissue using the Complete Genomics /MGI platform, as well as validation on the Illumina system as well. As Memorial University's genetics department, we are funded gratefully by the Marathon of Hope, Terry Fox Foundation, Canada Research Chair. Newfoundland and Labrador is obviously supportive of all of our research, and we'd like to make a generous thank you to the Biobank, Newfoundland Biobank, for its contributions. Next slide, please. Somebody, anybody? There we go. As you all are likely familiar, colorectal cancer develops in the colon or the rectum, often starting as a polyp in the inner lining. It's the third most common cancer globally. It's highly treatable when caught early, emphasizing the importance of screening, especially for those over 50. Risk factors include age, family history, inflammatory bowel disease, certain lifestyle choices, diet, processed meat, smoking, obesity, and excessive alcohol intake. Next slide, please. So the aim of our study is examining the mutation beyond the exons, regulatory elements specifically. Most studies focus on the exons with whole genome sequencing, and that focus on the genes focuses directly on about 2% of the genome, ignoring the other 98%. So by utilizing a combined multi-omics approach, using whole genome sequencing, RNA sequencing, and my particular project, ChIP-seq, we are able to focus on all the other areas of the genome and specific focus on enhancer and super enhancer formation following a mutation. Go next slide for me, please. Oh, thank you. So the enhancer can recruit transcription factors that either activate or repress the activity of the gene downstream. So by looking at somatic versus germline mutations, we can identify when that mutation occurs in the tumor versus the normal tissue sample. You go next slide for me, please. Perfect. So a seminal paper published about 10 years ago, 2014, showed that oncogenic superenhancers can be formed through somatic mutation, and this is in that non-coding area, so outside of the exon. This highlights the importance of our research focusing on that 98% that is not typically addressed in current studies. If you can go next slide for me, please. And to do that, we focus on functional regions of non-coding genome. As you can see in the slide here, unfortunately, I don't have control of the cursor, but as you can see in acetylation, we can find open active areas of the chromatin as opposed to areas that have been methylated, which is condensed or repressed chromatin, and unfortunately inactive and unavailable for us to evaluate. So we have chosen to use H3K27AC, which is an acetylated protein, and by using that we can actually evaluate the areas that are in open chromatin state and active functional use. Next slide, please. So our strategy of utilizing multi-omics, the whole genome sequencing, the RNA sequencing, and the ChIP-seq, my particular project, with that H3K27AC, with the gold cohort, we can take tumor samples, and by doing so, we're obtaining these samples from the Newfoundland Biobank. These are matched samples for a Newfoundland patient. It's a sample of the tumor itself as well as an adjacent normal tissue. So far, with our in-house sequencer, it's our next generation sequencer manufactured by MGI, which is now termed Complete Genomics, it's the G99 sequencer. We have been able to sequence 45 patients, which is 90 samples, one tumor, one normal for each. And I personally do the ChIP-seq. The other affiliates within the university perform RNA-seq and whole genome sequencing. And then our bioinformatics team combines that data from the whole genome, the RNA-seq, and the ChIP-seq together so that we can evaluate the activity of the genome in our specified locations. You go next slide for me, please. Up until now, the unfortunate thing has been that fresh or frozen tissue samples are very difficult to work with within the arena of ChIP-seq. There hasn't been a protocol available to provide reliable and functionally reproducible results until now. With our in-house sequencer, our access to the Newfoundland Biobank, and building on research that was performed by the Benicroft Lab in Singapore, and one of my collaborators and colleagues here, Khadija Rabbani, we've been able to take her cell line protocol for ChIP-seq and apply that to frozen tissue samples. Just yesterday, this was actually published in Current Protocols. As you can see, we have our cover page here with the abstract, which you can refer to later, and also our submitted artwork, which we will remain to find out if that's going to be the cover of the journal article. So I'll go through in some detail. If you can forward to the next slide, please. For my contribution, using our Complete Genomics G99, I follow our in-house protocol for the ChIP-seq and then follow their protocol from Complete Genomics for the library preparation with 14 PCR cycles. The samples are prepared for sequencing. We perform that sequencing in-house. The benefit here is our G99 is a mid-throughput sequencer, which allows us to optimize our sonication, our PCR cycles, our in-house ChIP protocol without the risk of sending it off to a high throughput facility and having poor outcomes and wasted time, money, and effort. So we're lucky very much to have that in-house sequencer available. And our in-house sequencer yields pair-end reads target depth sufficient for a robust H3K27AC peak detection. As I said, currently we have 35 patients of tumor and normal specific to the G99. and plans for 45 additional patients, hopefully by April of 2026. Now that we have reliable results produced by the G99, we've also collaborated with another clinical facility here at the Memorial University, which is CTG, to validate our ChIP protocol with an Illumina NovaSeq 6000, the SP flow cell. We're using the Takara Through-plex DNA sequencing kit for library preparation, and then that sequencing occurs in CTG, where the libraries are quantified, pooled, sequenced on the Illumina, and the pair end reads and analysis are compared then back to our G99. Currently, there are 10 patients that have been sequenced with the Illumina and analyzed. 6 are in process for sequencing in the next 2 weeks. These are patients, and an additional 6 by mid-December. If you can fast forward to the next slide, please. To give you an idea of what we're working with in the lab, and I was chatting with everyone while we were getting the seminar set up in the lab just now. What I'm doing is going down to the biobank, taking, as you can picture with the, well, you can't see my image, but if you can picture the head of a pencil, some of the tumor samples can be as small as the lead at the head of a number 2 pencil, and some as large as the eraser at the back of the pencil. So you can imagine that it's very difficult to get consistent tissue samples at such a small size and to be able to yield the necessary amount of chromatin isolation for our sequencing. You can see in the image on the upper left, that's a tumor sample. I'm processing the tumor samples on ice to keep them cold and preserved as well as possible, mincing up the tissue and utilizing, you can't see it here, but there are 2 different methods. We've been trying to validate cost-effective means of being able to do this in various locations throughout the world. We use a tissue grinder, which is a mechanical item with, if you can imagine, a bullet blender, which is basically a little test tube with plastic blades in it to finely mince the material. Or we've used a Dounce homogenizer, which is a lower cost, more potentially readily available option. It's basically a glass tube with a pestle in it that you can grind up the tissue manually. We then take our samples, go through wash stages, sonication to shear the DNA into predetermined appropriate lengthed DNA fragments, which we have found to be different between the G99 and the Illumina sequencing platform. Once the DNA is sheared, we then go through library preparation, quality control steps, and the sequencing. Next slide, please. So what you can see in this next figure, we take from the -80 the sample, prepare the tissue, mince, suspend the tissue, go through the mechanical dissociation. Next slide again, please. And then we utilize formaldehyde for cross-linking of our antibody with the proteins, cell lysis, purifying of the DNA, the sonication for the fragment lengths, QC steps to verify the length of the fragments, the antibody and DNA incubation binding, the wash step to remove the antibody and proteins and have just the remaining DNA fragments for sequencing. Next slide, please. Now here I have to acknowledge I'm not a bioinformatician. I focus on the wet lab, but our lab, with the help of the bioinformatics team, specifically Guillaume Tricnot, he was here as a visiting student for his master's internship just in the spring from Belgium. The analysis is focused on mapping the active enhancers in colorectal cancer in the normal and the tissue samples using our ChIP-seq data. Identify the enhancers that are differentially enriched, meaning that they are found in the tumor sample and not in the normal sample. Identify germline and somatic variants that are correlated to enhancers. Next slide, please. I'm trying very hard to stay on track here for our 12-minute goal. So within the data produced by the ChIP-seq, we find, if you look at the top of this graph, the normal and the tumor tissue, you see the enrichment in those 2 samples. And then in the zoomed in area in the middle of the screen, you'll see the bars that either show activity in one or both normal and tissue sample. What we're looking for is something that shows an enhancer activity in the tumor sample specifically and not in the normal tissue. If you can hit forward again, I believe there's, so in that further zoomed in area, you see the somatic mutation in only the tumor sample. So then we can identify those variants and go further into the study of those variants and how they may be interacting as an enhancer to a downstream gene. Oh, thank you. Little heart, isn't that cute? Go ahead and hit forward again one more time, please. So looking at an example of one of these results that we've gone on with our further analysis, we found 2 putative sites where they were predicted in the normal tissue sequence related to that enhancer, and 4 that were predicted in the tumor sequence. And you can see that those are different areas that are identified in the normal versus the tumor tissue. And with that focus, we can then evaluate the impact of that dysregulation at the transfer factor, and then the potential activation or repression of downstream genes. If you can go down one more slide, please. This enhancer analysis, actually, I'm not familiar with this figure. I don't know how that got in. But zooming in, you can look at the genomic region and the sample just to identify areas of interest. Our master student, Guillaume Tricno, went on, if you can go next slide, to perform a principal components analysis. And now this is not on our data, this is on published data, but is a fascinating potential trajectory for our research. Looking at the published data, we can find through the principal components analysis that the clusters don't fall out by normal and tumor. As you can see in the middle lower right condition, blue circle represents normal tissue, orange triangle represents tumor tissue. And what you might expect to find is that the normals cluster together and the tumors cluster together. But rather than that, the PCA actually indicated that they cluster by region of the colon and rectum, which could provide opportunity for us to evaluate ChIP-seq result and predict where the potential cancer originated from or is potentially going to originate from. So looking at these clusters, you can see the pink, blue, orange areas are related to regions of the colon and the rectum. If you can fast forward. So our overall conclusions and perspectives so far in this study, and this is ongoing, as I mentioned, is to characterize the enhancer landscapes in Newfoundland population of colorectal cancer patients, identify those variants within the enhancers that are differentially enriched, again, meaning that they are present in the cancerous or tumor sample and not present in the normal sample. Assess the functional impact of these enhancer-associated variants on the disease development within colorectal cancer. Discover epigenomic signatures that are unique to Newfoundland. So we do have the potential here to look at our population and compare those to other populations. Obviously, clinical data here is important to look at staging and location and heritage background. And then analyze the currently ongoing results as we produce them by our bioinformatics team. Go next slide, please. And I would like to acknowledge all of the contributors here. There are too many ACC members for us to list, but within the Benicroft lab, Matthew Dwyer, Gaston, Asma, Akinla, myself, Khadija, with a specific acknowledgement to Khadija Rabbani. The protocol was based on her cell line ChIP-seq protocol that she had developed years prior, as well as Guillaume Tricno, our former member, our master's intern, come from Belgium in the spring. Obviously the biobank here, Dan Luce, Luis Vide, John Toms, huge contributions from their consented collection of patient samples. And our Complete Genomics Canada, Ken has been an integral part of the development of our collaboration with the G99, Complete Genomics, and MGI platform. And unfortunately, I should have mentioned also Cindy Penny here at CTG, who is helping us validate all of our G99 results with that of the Illumina platform. And as you can see, we have a pretty great lab here, and a huge thanks to Twatty Benicroft for all of the wonderful opportunities he provides us. And with that, just bringing everything back to Terry Fox. Obviously, this funding and research opportunity is providing quite the productive future for all of us, I think. Thank you very much.
Thank you very much, Dr. Alloway, for that fantastic talk. I do apologize for the earlier technical difficulties, and a huge thank you to Peter for stepping in. I think my camera's still not working, but seems like you're hearing me, so that's fine. We have a few minutes for questions. I'd like to pass it over to my co-chair, Natalie. If Natalie, would you like to ask the first question?
Yeah, thank you. This one will go to Dr. Reiman, specifically about Dr. Amy Trottier's research that you presented. I'm fascinated by germline mutations. I'm a narcoleptic, as is my dad. And my dad is one of 5 siblings, born in France, all male. When I got diagnosed with breast cancer, we discovered that his mom, his grandmother, his aunts, his many cousins all have breast cancer too, funny enough. So when we were here, too late now, but we both got genetically tested and we both have mutations of unknown significance. Question, ... narcoleptic breast cancer, none of the above. So I was really fascinated to see that family tree, the family trees that you presented. And so my question is kind of twofold. What does this research mean to a patient or to the family members? And what are the next steps for her research?
Well, Amy's on the call, I think, so I don't know if you want to jump in, Dr. Trottier, but happy to respond on your behalf. Looks like she might be tied up. So the more the world, does cancer genomics and reads the genomes of various cancer cases and compares those to the inherited sequences that we all carry, the more genes are uncovered that provide some some contribution to our cancer susceptibility. And yeah, we know that doesn't explain everything, and that the genetics of many cancers are complex, and there's not always one gene for it, like there might be sometimes for, say, breast cancer, and that there may be some interaction between those genes and the environment. And so I think it'll speak to people's, understanding comprehensively people's susceptibility to myeloma or other blood cancers, and especially how to work with families of people who have been diagnosed with these diseases and who may feel that they are at risk, and who may be at risk, and to counsel them. Amy, do you want to jump in? I see you think you've got your hand up.
Yeah, thanks. Sorry, my mic was initially disabled, and so I couldn't jump in there right away. But thanks. Yeah, obviously this is quite a big passion of mine, and it's driven by the patients and families that I see that have these familial patterns. And I think, Tony did address it well, is that it is a complex matter in terms of these hereditary predispositions to cancers. And there are certainly some hereditary changes that we would consider a strong predisposition factor that make it quite likely that a person may develop these cancers in their lifetime. But there are also others, and this is more so the ones that we're still looking at and trying to identify, these more elusive ones where, as Tony was saying, it may be a combination of, it could be multiple co-inherited genetic factors that each one of those things is a small increase in risk. But if you add up enough of those small things, you get a big thing. And those are all of these things that we're looking at trying to identify. So leveraging the Marathon of Hope samples, we have all of this normal tissue in addition to the tumor. So much of the researchers you'll see here are using that to kind of get rid of those hereditary ones and just look at what's really just in the tumor, because the tumor is a mix of the cancer and the normal DNA. Whereas we're looking more at what is in that normal section. And can we find potentially new things or trends, like yourself, you say there's uncertain significance ones. And these are uncertain just because they haven't had enough study to know, are they actually linked with the cancer or are they not? So by doing this kind of work, we're increasing that database, that knowledge that we have on it. And that could change potentially this unknown significance to known significance. whether it be not causative or potentially causative. And the beauty with this and this phase 2 is that we're going to be able to take those results directly back to those patients and to their clinicians. And that's something that we do here locally, and we're developing it, a process to do it nationwide. So in some cases, we may be able to impact those people in the here and now. And then for many other cases, it's going to be in the future. Maybe we can't help that person, but maybe we'll have information for their children or grandchildren down the road.
Thank you.
Thank you so much, Tony and Amy, for that answer, and Natalie for that question. Unfortunately, that does bring us past the hour, so I think we'll have to wrap up. But I do want to give a big thank you to all the speakers and to co-chair Natalie. This was a fantastic seminar, and it's always, I'm a bit biased, but it's always wonderful to hear what's happening out east in the Atlantic region. So thank you so much to everyone. As Isabel said, this is the end of our series of consortium seminars, but we are starting a few other series, including a trainee seminar that will happen on December 18th. So you should have received a link to register in the email that you received as a reminder for this one yesterday. But we will advertise that further, and we'll have some more seminars in the new year as well. So again, thank you so much to everyone for attending and to everyone who participated, and we will see you soon.